So this is the catalase and oxidase test. They are two separate tests looking for two different enzymes. Both enzymes deal with respiration. Instead of anaerobic respiration, however, we're dealing with aerobic respiration here. So for both of these enzymes, we are looking at aerobic respiration. So that means that it's going to be oxygen that's the terminal electron acceptor. Now, these bacteria can either be strict aerobes or they can be facultative anaerobes, meaning they have an electron transport chain. They can use oxygen as a terminal electron acceptor, but they can use other things if necessary, and that's those nitrate reducers that we looked at in the previous video. So these are going to be using oxygen as a terminal electron acceptor. So we're going to start out by looking at the catalase test. So the catalase test, again, like I said, is looking for respiration. And during respiration, normal respiration with oxygen as a terminal electron acceptor, there are things called superoxide-free radicals that are formed. Now, these are normal intermediates that are formed in respiration. However, they are toxic to the cell. So the cell has to have some way of dealing with the superoxide-free radicals. So to do that, it produces an enzyme. The first enzyme that it produces is superoxide dismutase. Superoxide dismutase is made to neutralize the free radicals into hydrogen peroxide. So that's what that enzyme is responsible for, taking those free radicals and turning them into hydrogen peroxide. However, hydrogen peroxide is also toxic to the cells. It will kill the cell. So the cell has to have another enzyme that it produces to deal with the hydrogen peroxide. That enzyme is catalase. And this is the enzyme that we're actually testing for this test. The catalase takes the toxic hydrogen peroxide, that's what this H2O2 is, takes the hydrogen peroxide and neutralizes it into water and oxygen, which of course are not toxic to the cell. So some bacteria produce the catalase to handle this, and some don't. So we are asking the question, what does your bacteria do? So that's the catalase test. Testing for the presence of catalase. Catalase is responsible for taking hydrogen peroxide and neutralizing it into oxygen and water. The second enzyme that we're testing for is called oxidase, the oxidase test. And it, again, is dealing with respiration, aerobic respiration. And some bacteria produce this enzyme. The whole name of the enzyme is cytochrome oxidase, because these are going to be the bacteria that are using a cytochrome chain or an electron transport chain. So some bacteria produce cytochrome oxidase, and the oxidase is responsible for taking those spent electrons at the end of the electron transport chain and placing them onto oxygen to form water. So this enzyme is very, very important. It takes those spent electrons, places them on oxygen, which is the terminal electron acceptor, and makes water. If this enzyme wasn't there, the bacteria would not be able to do respiration. So our question is, does your bacteria produce this oxidase? It's going to give us more information about the bacteria. So, to test for the presence of catalase and oxidase, you have to do a streak plate. So here's my two streak plates that I did. Here's my bacillus. So this is one of the ones that we did, or that I did. And here's the E. coli. So I did an E. coli streak plate. Look at those beautiful isolated colonies. Aren't they so pretty? So the first test that we're going to do is catalase. So here's my catalase test. And remember, what's the catalase doing? It is taking hydrogen peroxide and neutralizing it into water and oxygen. So, all that is in this is hydrogen peroxide. It's the same hydrogen peroxide that you use at home on your bobos. So, what's going to happen is if the bacteria produces catalase, when I put this hydrogen peroxide on these cells, they're going to freak out. They don't like it. So if they have the enzyme catalase, they're going to break this hydrogen peroxide down into water and oxygen, or bubbles. So once I add the catalase, if I see bubbles, then that's a positive reaction for catalase. No bubbles would be a negative reaction. So I'm going to add my catalase to my bacillus. So you actually have to open the plate up and then put the catalase directly on the plate. So hopefully we can do this and you can see this easily. So you see those bubbles? Woo, look at all those bubbles. Alright, so that means that bacillus is positive for catalase. If you didn't get any bubbles, then that would be a negative reaction for catalase. Pretty simple, right? So bubbles, positive for catalase, no bubbles, negative for catalase. And remember what that catalase is doing. Next is the oxidase test. Remember, this is testing to see if the bacteria produces that enzyme that turns the oxygen into water. Okay? So, to do this test, I'm using an oxidase reagent. I'm not going to tell you what it actually is because it's a really, really long word and y'all don't need to know that. All you need to know is it's the oxidase reagent. And I'm going to take my sterile cotton swab and I am going to get a colony on the end of my sterile cotton swab. Now it's like a yellowy color. I don't know if you can see that on the sterile cotton swab. That's my colony. Now I'm going to take my oxidase and put it directly on the colony. If this is positive for oxidase, the colony itself will turn purple. It's a dark, deep purple. So I don't know if you can see that, but you see that colony is still yellow on there. No purple color. See? Colony is still yellow, no purple color. So that is negative for oxidase. So E. coli is negative for oxidase. So it does not produce oxidase. Now I'm going to do the same thing, but with my bacillus. So I got a new sterile cotton swab. Got my colony on my sterile cotton swab. It's kind of hard to see because it kind of blends in with the Q-tip. And I'm going to So we're going to give it a couple minutes to change, but you can already see that beautiful purple color that those colonies are turning. So just to compare, here's my E. coli. Clearly the colony is still yellow, no purple color. And here's my positive result. Nice, dark, deep purple colonies. Okay, and that's what we want to see for our positive result for our oxidase. So oxidase positive is purple, negative is no color change on the colonies. Looking for the presence of two different enzymes, both dealing with respiration. Catalase is responsible for neutralizing hydrogen peroxide into water and oxygen. Positive result, of course, is bubbles. Oxidase is responsible for placing the spin electrons onto oxygen, making water. Positive result is a nice dark deep purple. Negative result is no color change on the colony. Okay.